Hello, everyone. Welcome to today's webinar, What's New in Research. I'm Jenna Mvalo, and I'm the National Director of Programs at the MGFA. Before we get started, I want to just go over a couple of housekeeping items with you. Today's session will be recorded and available online following the presentation. If you would like to use closed captions, you can do so by clicking on the CC button located at the bottom of the Zoom menu bar and select English as your language. We ask that you submit your questions in the Q&A box located at the bottom of the Zoom icon, and we'll be able to answer those during the Q&A segment. Now I'd like to thank our sponsors for today's webinar, NMD Pharma, and hand it over to Tomas, who's going to introduce the rest of our speakers today. Welcome.
Thank you so much, Jenna, and a big thanks to MGFA for facilitating this webinar. We are very, very excited to be here and to present to you about NMD670, a novel first-in-class CLC1 inhibitor that is being developed for myasthenia gravis. My name is Thomas Grønnebæk, and I work as a principal clinical scientist for NMD Pharma, and I've joined here with my colleague Vera Kirsova, our medical director, and Professor James Howard, who will initiate the webinar with an introduction to why it's relevant to continue to pursue new treatments for myasthenia gravis. Thank you so much, and looking forward to the discussion today. Over to you, Professor Howard.
Thank you, Tomas. Welcome, everybody, to this webinar. And if I could have the next slide, please. So why do we need new therapies? You know, if you read the news and all, the myasthenia space is rapidly expanding. We've had multiple drugs approved in the last few years. And do we really need anything new? And I will submit to you that yes, we do. When we look at the burden of disease and the burden of treatment on patients, it's still pretty outstanding. Patients complain of the fact that the drugs they're taking don't do what they're supposed to do. They're not as strong as they would like. They have adverse events that are intolerable or make their quality of life bad, if you will. One of the outstanding things or shocking things that came to my attention a few years ago was the fact that many patients with myasthenia are unemployed. For instance, in Thailand, more than 50% of myasthenics cannot work. In Japan, it's over a third, and another 5 to 10% have a reduction in income such they can't care for their family. In Australia, the number of unemployed runs in the range of 40%. Japan and other countries, it's quite similar. In the US, because of our fractionated healthcare system, we can't get good data, but we know that absenteeism in jobs in which myasthenics are employed is fairly high. In addition, if we can go to the next slide, what we're finding when we do our clinical trials, and this is a list, it's not complete by any means, but when this slide was put together a few months ago, this is the data we could get. If we, as you know, trials are blinded. Half the patients or a percentage of the patients get the real drug, the other half get a placebo, and then at some point in time, they cross over and get the real drug in what's called open label. In the blinded phase, if we only look at the blinded phase of the study, and then ask the question, how many percent or what percent of patients don't respond to the drug we're giving them, the figures in the right-hand column are astounding. The lowest is 14%. The highest is 48%. Nearly half the patients in this trial did not respond to the drug. And for the most part, it's in the range of 30+%. And that's telling us something. It's a population we don't really explore, but I think it points to the need that despite our best efforts and despite bringing new therapies to the bedside for you, we still have lots of patients who really don't respond to what we're doing, and hence the search for new tools. What you're going to hear about is a very unique compound, NMD670, which I'll let the experts go into more depth, but it offers a niche at how we can be able to potentially help individuals with myasthenia. And with that, I'll turn it over to Vera.
Thank you very much. Next slide, please. Thank you for a very good introduction and setting the scene. And before going into all the technicalities and details about this compound per se, I'd like to stop and reflect, because it's probably one of those webinars you're used to hear about neuromodulation, neuroinflammation, about many other things, but not on specific skeletal muscles. And that's what we are planning to talk about. Why add on muscle targeted therapies? Why? It's as simple as that. Skeletal muscles is basically half of the mass of the human body. And interestingly, the first thought that comes up to mind, yes, of course, we all need muscles. Muscles are there for mobility. But let's also not forget that it's not only the movements. Muscles are not only there for mobility. It's also a great endocrine organ. Muscles play important role in our metabolisms. It's the largest energy consuming organ, and it's largest store and reservoir of metabolites, including glucose and protein. And what is extremely important, and I think it echoes nicely what Professor Howard also just mentioned about absenteeism or incapacity to work, incapacity to function, muscles are that very organ that is directly linked to longevity and to the quality, to the length, and to the quality of life. Next slide, please. What we know now is that unfortunately, skeletal muscles dysfunction can be seen in multiple rare and not so rare disorders. What you see on this slide is the representation of different diseases. Some of them are very well known as type 2 diabetes or multiple sclerosis, sarcopenia. Some are rare, like myasthenia gravis, which you know very well, but it's not so very well known by entire community, myotonic dystrophy, many others. And what we also know right now is that in all these pathologies, different types, it's either neuromuscular junctions that suffer, or they can be classified in skeletal muscle ion channelopathies. But without even getting into details and medical classification, I'd say it's just a bit of a puzzle for all of us to stop and think in how many disorders muscles are affected, and how important it is to get to new, more efficient treatments for the conditions where it's such a vast unmet medical need. Next slide, please. And here's the story of the company that was born in university environment in Aarhus, which is the second largest Danish city, and a city with a very big history and culture of research, a lot of students, one of whom, as you see on the picture, named Thomas. Yes, we have 5 Thomases or even more in our small company, but this is Thomas Holm, who is a scientist per background and the current CEO of our company, who many years ago been interested as a PhD student, as a postdoc afterwards in muscle physiology, and had this beautiful idea on observing and describing the CLC1 channel that we're going to talk about it later, starting a company that 10 years ago looked like this, just a small group of people. Next slide, please. And that grew in 10 years in this group of individuals. So this is the most recent phases of the company that we are now, NMD Pharma. from our offsite in Denmark in May, where we celebrated 10th birthday of our company, and as well the progress that we've made into the clinical stage biotech, developing novel treatments for 3 neuromuscular pathologies at the same time right now. Next slide, please. So who we are. You saw faces. You see our purpose now. What we are working for is trying to do our best with all our different backgrounds, professionally, culturally, enabling better lives through novel therapies that restore skeletal muscle health. And as you understand now, we are specifically focusing on therapeutics for neuromuscular diseases, having one lead program with the molecule that Professor Howard mentioned, NMD670. And now we are studying how it can improve muscle function in patients with myasthenia gravis in phase 2 study. Our company has strong academic foundation and unique capabilities. It's developing actively with growing financial needs, but growing as a company, hopefully very soon progressing into the later stage clinical development. Next one, please. And before getting into details of the clinical development, I'd like to take this time and go into some details, hopefully explaining them as simple as possible. And if not, we can always welcome your questions on what is actually this compound doing in the muscles. But before going into treatment, let's talk about motor system and how it works. So skeletal muscle fibers, we know in red here that you see, cannot really demonstrate it with my pointer on the slide, but I'll explain it with my hands. So skeletal muscle fibers are activated by signals called action potentials that basically travel from the brain to the muscle fibers. The power or the muscle force is controlled by the action potential frequency, how often do they come, and by the number of the activated muscle fibers. And it's important to remember that motor neurons and muscle fibers are highly adaptable based on the way they're used, which means they can be highly adaptable, active, and muscle can grow if they're used, and they're as well, in a negative sense, adaptable to inactivity, and that's where we have the muscle wasting. Another important fact to remember is that in this crosstalk, in this relationship, it's a real relationship and a dialogue. And it's always adaptation involves both nerve and muscle that communicate to each other by releasing trophic factors or nutrients, we can say, from the nerve side and the muscle side. Next one, please. This system is extremely well designed. That's why during our entire life, it certainly helps muscles activations, that enables our dexterity, helps us, keep the balance, corrective movements. But unfortunately, as any other well-designed machine, even the best designed machines have some vulnerabilities. And that's what happens here, is that the neuromuscular junction, so the place, this that you see on the muscle with little circles, the connection in between nerve fibers and muscles, sometimes is affected in the range of the disorders, and not only myasthenia gravis. Next one, please. So efficient therapies and safe therapies to improve neuromuscular transmission at the level of neuromuscular junction and muscle activation could potentially address this unmet need in neuromuscular diseases. Next one. And next one, please. So here it goes to the discovery and the description of our CEO and scientist in heart, Thomas, when he was working on the CLC1 chloride ion channels, describing that this is an ion channel specifically expressed only in skeletal muscles along the entire muscle fiber membrane. And what is it doing? What is its physiological role if it was found in the normal, healthy muscle? Well, its role actually is functioning as a brake for signal transmission from nerve to muscle. So saying it simply, in the muscle of in the healthy muscle, that's exactly the channel that will play the role for the muscle not to get overactivated, over-contracted. So by finding this, discovering this role and this function, the thought came, okay, if that's the role in the healthy muscle to not get overactivated, and we know that there's a number of disorders where the muscles need this help, why not to try to block this channel and basically give an opportunity for muscle to contract better? Next one, please. And that's from hypothesis and idea through many different preclinical models, what we came to learning, understanding, and getting to know even more, that this mechanism may help in disorders that are classified as presynaptic dysfunction. So basically where we know that there is a reduced release of acetylcholine, or in the disorders that are thought to be a postsynaptic dysfunction, as myasthenia gravis, for example. Next one. So CLC1 inhibition restores muscle activation. And on these images, what you see is the muscle on the upper panel. It's the muscle with compromised neuromuscular transmission. So basically the little circles, these are deficient or non-active dysfunctional neuromuscular junction. And when the NMD670, which is the molecule's name and which is the CLC1 inhibitor, or the molecule that blocks this channel, is administered. Can you click the animation, please, Jenna? That's what you see on the upper panel. And here we go. After the CLC1 inhibition, what we know is that it restores transmission and muscle activation. Next one, please. And as many of you are aware and been following a number of research webinars, when the ideas come in some preclinical settings, very often we study it first on the muscles, and then we go through different animal models to test the hypothesis, to see whether it's safe, to see how it works. And that's exactly what we've been doing. Showing at this panel on my left, on your right, that if we look at the action potential in the healthy muscles, we know how it looks, and we know that the muscles in certain rodents with myasthenia gravis don't have this capacity. However, if we administer the molecule NMD670, then they recover the capacity to contract and generate force. We also demonstrated in other different models a similar action on the improved breathing, so action on diaphragm, and increased survival in animal models. So all in all, before giving the word to my colleague Thomas, who will introduce all the clinical part, what I'd like really to bring to your attention is that we are talking about a unique molecule that acts fast and can not only help the muscles to function better and generate better strength at a shorter term, but if it's administered at enough long term, then it also improves this communication, this relationship between nerve and muscle at the neuromuscular junction, which ultimately at a longer term may also lead to the better muscle function, to the long-term benefits that all the patients are looking for. Thomas, to you.
Thank you so much, Vera. So we'll now shift gears and talk about NMD670. NMD670 is the first ever CLC1 inhibitor to be tested in clinical trials in humans. It's formulated as a tablet and it's supposed to be taken twice daily with approximately 5 to 8 hours apart. It works exactly as just described by Vera by enhancing the communication between the nerve and the muscle. And we hope that this will increase muscle strength, power, and endurance, and this is exactly what we are studying in ongoing clinical trials. As you will see on the left-hand side of this slide, NMD670 is currently being tested in 3 phase 2 studies in patients with myasthenia gravis and 2 other neuromuscular diseases. But prior to initiating these ongoing studies, the initial safety and efficacy was first evaluated in healthy volunteers and in a smaller study with individuals with myasthenia gravis. Next slide, please. I'll go into a little bit more detail with the results of the study in myasthenia gravis that was already completed. So this study involved a total of 12 individuals with myasthenia gravis. They all had autoantibodies against the acetylcholine receptor and generally mild disease. The patients were randomized to receive either a dose of NMD670 at 1200 milligram, 400 milligram, or placebo. And they were then washed out of the drug for a total of 7 days before then crossing over to receive one of the other treatments. So all patients received all possible treatments. Before and after the single doses, we did some tests to measure the effect of the compound. And this test involved performing the quantitative myasthenia gravis scale, which is a scale that I'm sure most of you is very familiar with. It essentially assesses muscle strength and function at different muscle groups. You will see the most important results to the right-hand side of the slide. The red line is indicating what happened in those patients that received placebo. You will notice that there is a clear trend that the line goes up on the graph, and this indicates worsening of symptoms over the day consistent with fatigue development. In those patients that received 400 milligram and 1200 milligram of NMD670, there were improvement in the symptoms noted as a downward trend on the scale. And when we analyzed this statistically, there were statistically significant different effects between the treatment groups, with higher treatment effects noted in the active treatment versus placebo. This, of course, provided very exciting proof of mechanism for this novel mechanism of action, and then further motivated additional trials in myasthenia gravis. Next slide. Another very important piece of result from the completed study in myasthenia gravis patients is the safety and tolerability data. This table here is summarizing the most important safety and tolerability data. In general, NMD670 was safe and well tolerated. There were no serious or severe side effects reported, and the incidence of side effects were similar between those that received NMD670 and those that received placebo. Next slide, please. So on the basis of these promising safety and efficacy results, NMD Pharma decided to move this drug forward into the next stage of clinical development, into what is called phase 2. This is a larger study that tests the compound for a longer treatment duration as opposed to the previous trial, which was a single dose study. Here we are studying the efficacy and the safety and tolerability for a total of 21 days. A total of 84 participants will be included in the study. 36 of those will be randomized to receive placebo. 6 will be randomized to receive a low dose of NMD Pharma, NMD670. 6 will be randomized to receive a mid dose of NMD670, and 36 patients will be randomized to receive a high dose of NMD670. The study will involve both participants with antibodies against the acetylcholine receptor and MuSK. There will be twice daily dosing for a total of 21 days. The most important endpoint of the study is the quantitative myasthenia gravis scale, which was also the endpoint that was used in the previous study. But the study will also include other measures of efficacy, including myasthenia gravis activities of daily living and other scales that will be used to ascertain how the drug may benefit the patients. Next slide, please. Let's have a little bit closer look at who may be eligible to participate in the study. So to be eligible to participate in the study, you must be between 18 and 75 years of age. You must have a diagnosis of myasthenia gravis with autoantibodies against the acetylcholine receptor or MuSK. You must be able to swallow tablets as the drug is formulated as tablets. These are not complete list of study requirements, and if you are interested, the study doctor will review all the requirements with you and determine whether you meet the requirements or not. A total of 84 participants with myasthenia gravis will be participating in the study in approximately 10 countries, and we'll have a closer look to that in a second. Next slide, please. The study is divided into 3 periods. There is an initial screening period, which is up to 4 weeks. There is a study treatment period, which is 21 days, where the participants will receive the medication or placebo. And then there is a follow-up visit after treatment is discontinued. During each study period, there will be 1 or more visits at the clinical center. The screening visit, the visit at day 14, and the follow-up visit, it will last about 2 hours, while other visits will last for approximately 3 to 5 hours. Before any study-related tests and procedures can be done in this study, you'll be asked to read and sign an informed consent form. And then the study will begin with a screening visit where the study doctor will review and evaluate whether you fit the criteria for the study. And forward, please. If the study doctor decides that you meet all the requirements to enter into the study, you'll be randomly assigned to receive one of the 3 active doses or to placebo. Next slide, please. So as said, the study will be conducted in 10 countries, and these includes the US, France, Denmark, Spain, Italy, Poland, Belgium, Serbia, Georgia, and the Netherlands. The first patient was enrolled in June last year, and the enrollment will continue well into the next year. Next slide, please. Let's have a little bit closer look at the US sites that will participate in the study. We have a site in Missouri, in Columbia. We have 2 sites in California, one in Orange and one in Carlsbad. We have also 2 sites in Florida, one in Boca Raton and one in Tampa. We have a site in Aurora, Colorado, one in Portland, Oregon, one in Chapel Hill, North Carolina with Professor Howard. And then we have a site in Augusta, Georgia, and in Memphis, Tennessee. If you're interested to participate in the study, you may contact the site directly by reaching the site contact site study coordinators using the site contact details in the right-hand column of the table. You may also reach out directly to NMD Pharma using clinicaltrials@nmdpharma.com, or you can reach either Vera or myself directly in case you have follow-up questions. Next slide, please. So what I want to finish off with here is just to say, if you are also looking for a drug that may reduce muscle fatigue, that may improve muscle strength and endurance, that is an oral treatment that works differently than other medications in this field, fast acting, and a tablet to be taken at home with no major side effects on the gastrointestinal tract, then we hope that we are developing a medication for you. And we hope that we have sparked your interest. Before we finish off, I'll hand it back to Vera, and then we'll open the floor for questions.
Thank you very much, Jenna. To the next slide, please. So I've started from the history and from the science, but I'd like to finish by giving back to community and sharing with you, and especially being grateful to MGFA for giving us an opportunity to do it, is that not only NMD Pharma is the clinical stage biotech where we do develop novel molecules, but what we're also doing is that we are learning from the community and trying to give back. And at the last meeting at the last patient conference in Arizona, I partnered with patient with myasthenia gravis, Deborah Vick, whom you can see here on the picture, in her wheelchair and her dog. And we created an adaptive yoga program, which is called Breathe, Move, and Thrive, Yoga for Everybody. And I see already among the participants of today's call, there are some of you who's been at this meeting and who participated in the initial sessions. And if we go to the next slide, Jenna, please, and the last one, what I'd like to bring to you is as the message is join us for more, because one thing is having your medications moving and already living your challenging life, including as much movement as possible. And the other thing is also joining us in the community for the program that's been really developed together by me as a doctor and as a yoga therapist, together with a patient who knows the everyday challenges, who very often finds the words during our yoga sessions to help, to encourage, to overcome, or on the contrary, to take a little bit of rest, and who knows better than any other doctor, because it comes from her everyday experience. It's free, it's for all of you, it's adapted to all of you, of us. And I know that when we made several sessions, especially for the Southern California support group and some others, we had a very positive feedback. So the more of you can join, the more of you want to call and have more, details on this. Happy to answer. And now the floor is open to the questions on the main topic of today's webinar. Thank you.
Thank you so much. We do have some questions, so we'll dive right in. This first one is, are any of these studies looking at seronegative subpopulation? This is particularly interesting since this drug does not act on AChR system.
So this initial study that I talked about and also the ongoing study did not and are not enrolling seronegative patients. We very much hope at some stage to be able to conduct clinical trials in seronegative patients once we hopefully have seen that the drug works in other forms of myasthenia. So it's definitely the goal, and we hope to be there one day.
Thank you. This next question is, how long will the treatment endure? Is it a short-acting tablet?
So that's a very good question. My colleagues can also chime in here, but it's very true that you can say initial effect of the drug is very rapid, and we saw that in the clinical trial where we saw improvements in symptoms within hours of taking just one dose. However, we have also seen that, and this is especially data from preclinical studies, that when we give the drug for a prolonged period of time, there may be other things going on that could lead to more sustained effect with this molecule. But this is something that we are still looking into. Vera, I don't know if you have any additional thoughts.
Yeah, thank you very much, Thomas. I completely agree with your explanation. What I'd just like to add is quite often when we start introducing our molecule, the question would come up. It's fast acting, so basically it's like Mestinon, but just without secondary effects. And I think it's also important to understand the nuance is that it's a fast acting, but with these 2 tablets that we are administering now during the day, it covers almost 24 hours. So it is fast acting, but it's not so quickly releasing from the system. And as Thomas also mentioned nicely in his presentation, contrary to the other molecule that many of you are used for its fast action, it doesn't have this important side effects either from gastrointestinal system or hypersalivation. So we still studying and learning more and more about it, but the way we understand it right now is that it covers, these 2 tablets are covering your entire day, and if taken at a longer term, then probably there is a certain add-on action.
Thank you. This next question is, what happens if you intensely exercise on this drug?
That's an interesting question that we have not studied. But a very, very, you can say, scientifically interesting question that at some stage we would like to be able to talk to with actual data.
Thank you. I think something else that falls along the lens of the exercise question is during the therapy or exercise, if a patient experiences muscle fatigue, should they push themselves or limit their movement?
I think this potentially could be a question for you, Professor Howard. Do you have any thoughts on this topic?
Thanks, Thomas. We know, and all of you know, that as you exert yourself, you become weaker and have more fatigue. And the rule of thumb that I've always used with my patients is that it's okay to push it, expect to pay the price, and have fatigue or some weakness thereafter, but you'll recover from that. And if the physical activity makes you feel better and helps your muscles adapt because they've been inactive for a period of time, that's potentially good. However, under no circumstance should you push to the point of pain, and you should stop if any pain occurs and rest. The pain is telling you something that may lead to significant injury to the muscle. So if you're able to push and you believe it's helping you, fine. It's not going to have a long-term impact. You may pay the price the rest of that day, but pain occurs. You must stop for your own safety.
Thank you. This next question is, will study participants have to discontinue Mestinon?
Mestinon can be, in general, continued for the duration of the study. However, there are certain visits where we will ask the participants to refrain from single doses of Mestinon, as this can potentially interfere with some of the assessments that are being performed in the study. But in general, in the duration of the study, the participants can continue on Mestinon.
Thank you. Can recently diagnosed people participate in the study?
Yes, they can.
Thank you. This next question is, with swallowing often being affected, have you considered alternative modes of administration? And if so, why is the tablet form used?
Excellent question. And yes, we not only considered, but we're also working on finding alternative ways of administrating in liquid formulation. However, let's say historically also, there are processes and procedures solve the issues of solvability and so on and so forth, and the way to keep the molecule in the best possible way to go through the GI tract and then get distributed. That's how it historically happened. And also there's been, in full transparency, I think there's been a number of discussions as well, even with the regulatory agencies who accepted that it's still an acceptable form in the at the level of the severity of the disease of patients whom we are currently enrolling in the trials. But to your question, we are absolutely empathizing and know that that's the difficulty, and that the liquid formulation might hopefully be the next step of the clinical development for the larger program.
Thank you. This next question is, would this drug eliminate both an immunosuppressant and Imuran or prednisone?
You mean eliminate the need for these therapies? Maybe that's
So.
what was meant. Yeah. So NMD670 is being developed as an adjunct therapy to the currently available therapies, which means that it can be taken in principle in combination with any of the already used therapies.
Thank you. This next question is, what effect will CIC1 have when there are collagen and ligament issues, such as with HEDS?
I am not totally sure I have an answer to that. Vera, any thoughts on this topic? Mr. Howard, you're also very welcome to chime in.
I don't think there would be any effect on Ehlers-Danlos or other hypermotility disorders of the joint and ligament. This is only expressed in muscle membrane itself, nowhere else. So I would not anticipate any problem or any interaction with hypermotility disorders.
No, I don't think so either.
Agree.
Thank you. This is a question and a comment. I'm from Michigan. Is there any chance that I can get to participate in this trial?
Absolutely. And I think that was also the intention for us. A very good question. Thank you very much. For all other who are here during the webinar from different states. So we selected, indeed you saw the map with a certain number of sites, but it doesn't mean that you need to live close to the site. So the idea is, from now on, you've seen us, you've heard us, you can contact either of us, and then we can discuss, which site location is easiest for you for travels, taking into account work, all the other obligations. And then then we'll put you in contact with the study coordinator at that particular site. And you can certainly plan for the screening visit and all the travel expenses. And even if you need to stay a night in the hotel near the site, it is all reimbursed within the study participation, because that's really the idea. We cannot have clinical sites all over the country, but we are trying to do our best and to facilitate for the patients, participants, that the travels are arranged and that you can participate in this trial. So just reach out and we'll let you know how to do it in practical terms, and you certainly can participate.
Wonderful, thank you. This next one is, is there anyone over 60 that has different side effects than a younger person when taking this drug?
Not that we know of, but what we should say about NMD670 is that it has only been studied in a relatively few participants at this stage of development. So the continued safety monitoring is something that that of course is very important, and is something that we'll be looking into at each stage of development. But so far, we have not seen any age-related side effects.
Thank you. This next question is there an expected time frame for approval and release for the GMG patients in the United States?
I mean, there are hopes and goals, but it's drug development, and there are many bumps along the road. So we cannot make any promises that this will ever come to market. If everything goes well, maybe around 2030.
Thank you. Vera, were you going to say something else?
No, completely agree that we cannot promise anything, but that we are trying to do our best. And also, if working with MGFA and other patient organizations on trying to spread the word, increase the awareness, it's all the joint effort, basically, to, despite what the community thinks, that there's been many recent approvals. And now what we heard from Professor Howard is that even though many new drugs are available, there is still a high unmet need. So this is still an early stage of development where every patient who is motivated enough is needed to advance this for the community, so that the next steps become possible, those as larger phase 3 that are needed for approvals and go to market.
I think it's important, Vera, to expand on that. In terms of drug development, you heard initially about early phase, preclinical with animal models. A signal was found. We went to healthy volunteers, phase 1 trial, where there was, again, a signal that this is potentially effective. And from that very small group, we then quadruple the size for what's called a phase 2, which we're in the midst of right now. And this is where we hammer out appropriate doses, get a better idea of what the potential adverse effects or side effects are, and is there a true signal? And seeing that, we then move on to what's called a regulatory trial or a phase 3 trial, which we have an even larger number of patients. MG trials somewhere between 150 to 250 patients are then enrolled. And if that works out to be well, then we go to regulatory agencies and seek their approval to market the drug. It's a very long and arduous process. The key aspect in all of this is safety, 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 first and foremost, all the way down the line, followed by, is it beneficial? Is it going to be of help to the patients that we're trying to use it in? So when we hear about these new compounds, we're very eager to be able to have it. But I tell you, it's a very long and arduous process. And we gnarl our teeth in get frustrated because we want things to move faster, but oftentimes it moves at the will it wants to move, and we have no control over it.
Thank you for that context. This next question is, as not all my muscles are affected at the same time, what have your patients seen with their intact muscles or not that affects the muscles?
What we can say at this stage is that we have seen beneficial effects across a range of different muscles. The QMG ascertains muscles like facial muscles, eye muscles, peripheral muscles. And across these different muscle groups, we saw trends of beneficial effects that are now being, of course, confirmed in those larger studies that Professor Howard also talked to. But in principle, should work in any skeletal muscle.
Thank you. The next one is, are there plans to study this drug for any other neuromuscular diseases?
Not only plans, they are ongoing. So there is a very similar study to the one that is currently being conducted in myasthenia gravis patients, also in Charcot-Marie-Tooth disease and in patients with spinal muscular atrophy. So already 2 additional studies ongoing in 2 other neuromuscular disorders. If these studies are positive, it's even likely that the drug could be expanded to other neuromuscular indications as well. And the reason for this is that there seems to be this, you can say, common or shared characteristic of many neuromuscular disorders that Vera also talked about, this failing communication between the nerve and the muscle. And this is exactly what this new drug potentially can address.
And if I may add also, this echoes Professor Howard's line on safety, 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 is when we also answered most of your questions about safety. Indeed, in myasthenia gravis, we had this small 12 patients initial phase 2A study and the current ongoing studies. But because we are currently working on 3 programs in parallel, we're also getting a lot of safety data from other disorders like SMA and Charcot-Marie-Tooth, where populations are also quite different ages, where you can also imagine that they have different concomitant medications. And so far, we really see that compounds being well tolerated and the safety profile is good.
Thank you. This next one is, as Dr. Howard mentioned, approximately 30% of patients don't respond to novel therapies in clinical trials. How is NMD670 faring in this phase 2B study?
That's a very good question. And we don't have the answer to that right now because the study is still in the blinded phase, which means that we as the sponsor and also the sites and the investigators, they do not know which patients received the active treatment and which received placebo. So we cannot answer to that question at this point in time.
Thank you. And then this last question is, just so I'm understanding this, have you not seen any bad side effects so far?
Good question. I think one thing that is relevant to talk about is what happens when you give the drug at very high doses. This is something that is done in the very first studies that are conducted in healthy volunteers to see how high can you go in the dose before you start seeing side effects, or in this case, exaggerated on-target pharmacology. So actually too much of the effect that you are expecting of the drug. And with too much pharmacology, too much of an effect, the muscle develops what is called myotonia, which is a phenomenon where the release times from a contraction becomes prolonged. The muscles becomes stiff, but this is something that we have only seen at much higher dose levels than what is currently being tested in the ongoing clinical studies, and also at doses much higher than is eventually intended for marketing if the drug makes it that far. But as Vera said, as Professor Howard also said, we are in the very early stages of clinical development, and we are continuing to monitor for side effects. This is an obligation in drug development.
Thank you. Well, thank you all so much for your presentation today, and thank you for the information. Thank you to everyone for participating today. We look forward to seeing you on another one of our webinars, and I hope that everyone has a great rest of their day. Take care.
Thank you.
Thank you so much, and thank you for the great questions.